Some of you do remember. So we talked about mucous membrane being part of the GI tract, right? And mucous membrane also you will find it throughout the respiratory tract, urogenital tract. So it's pretty much similar type of structural or structural difference or similarity you will find. So when you look at, okay, we have to finish the immunology in today's lecture. Then also afternoon I supposed to teach antibiotic resistance. So it might happen that I may have to continue discussing immunology until this afternoon. Then antibiotic resistance may be tomorrow. Okay, so there will be a little bit shuffling and rearrangement, but nonetheless we'll finish everything we plan to do it by Friday, right? As the goal before you take your exam. So few things just to point out the mucous membrane. As I said, we have a pattern recognition receptors, TLR, and these are like a recognition molecules. The most commonly we call it also PAM. Sorry, PAMs, right? Pathogen associated molecular patterns. So mucous membrane should be able to recognize microorganisms, whoever in that vicinity or in that location, and your immune system should be able to communicate through that pattern recognition molecule, right? So I will explain a little bit more as you go along. And then I also mentioned that the antimicrobials, we had quite a discussion there, that Paneth cell is one of those epithelial cells I showed you earlier that secretes a lot of small peptides, bacteriocin-like antimicrobial peptides. Those are not antibiotics but antimicrobial peptides, and it essentially try to inactivate pathogens or microorganisms in that environment. So that also contribute to the immunity. And of course this is some of the examples of antimicrobials: lysozyme, cryptine, this is another one. This is about 35 amino acid long peptide. Then you will also find, I mentioned about defensin. You heard about defensin? Macrophage, neutrophil, they have these antimicrobial peptides, and those are also very effective in controlling pathogens. And then goblet cell, of course, very important, right? Mucous membrane, without mucus, it doesn't work that well. So you need to have a mucus secretion, and we mentioned that goblet cells, they secrete mucus. Okay, and we also started discussing a little bit about the epithelial barrier damage, right? Leaky gut, and I'm going to talk a little bit more about that as we go along. So essentially, you are disrupting the epithelial barrier in the gut, and so we have microorganisms in the gut, right? At large quantities, large numbers, and you don't want them to cross our epithelial barrier to get to the blood circulation. So that would be definitely a negative impact on our health, including joints and the brain. Okay, so the barrier effect can be in the lungs or in the gut, right? And so as the immunogen or microorganism passes through this system, it activates immune system. Definitely you don't want the immune system to be overactive, and consequences of overactivity is that they are going to produce a lot of inflammatory cytokines, and inflammatory cytokines are not desirable. At low level they are desirable, but as the concentration increases, that necessarily has negative impact on health. Okay, mucus, a little bit about the mucus. Mucus is a glycoprotein. Mostly it's actually carbohydrate, right? Let me see if I have a structure of it. No, I don't have. I thought I had one. No, I don't have the structure. So mucus is actually large complex. Okay, it's carbohydrate, but then core of the mucus is called protein called mucin. Okay, and with a hydrogen bond, it forms complex structure. So it's a jelly-like structure, right? And so obviously that's been secreted by the goblet cell, and it has a lot of functions. Instead of talking too much about it, because it has carbohydrate, I mentioned that even if you don't have food for a while, you can still survive. And you survival means your microorganism in the gut. So they can feed on the mucus to survive for extended period of time. Okay? But the mucus, because it's a gel-like structure, it holds all those antimicrobial peptides. A lot of different molecules are present in the mucus. So therefore, mucus is extremely important, right, for our health. So healthy gut means we need to have a healthy mucus layer. Okay? And these are some of those descriptions of the mucus. The molecular weights, like I said, in the millions. Okay? And these cells die pretty quickly because of the programmed cell death, and they will renew every 5 to 7 days. And other aspect of the goblet cell, they respond to microorganisms very well. Whether they're pathogenic or non-pathogenic or probiotic organisms, they will play accordingly. So some pathogens may increase the mucus secretion, whereas some pathogen may decrease the mucus secretion. Okay? Because that would be advantageous for them if they reduce the mucus secretion, so that they can be invasive, they can go through the system. And sometimes they may stimulate more mucus secretion. So therefore, they may have a upper hand in competing with the natural microflora. Okay? So they do a lot of weird stuff. Okay? But nonetheless, all happens. So the mucus, again, if you study a little bit more, there are 18 genes actually control the mucus secretion or mucus synthesis. One of the genes is called MUC2. So a lot of times we look for the MUC2, which is the secreted form of the mucus. Okay? And we can monitor the secretion of the MUC2 when you are studying, say, effect of probiotics or effect of pathogens or effect of toxins. So these are the way we could monitor it. If you look at the mucus, there are two types of mucus. Okay? Again, one is called sticky mucus. Sticky means it attaches to the epithelial cell. And the other one is called loosely attached. And so this is in the chicken actually, that's the advantage, that's the anatomical classification we use. So this is the front part, like esophagus equivalent to humans, and then the antrum, and then this is almost like a stomach, right, in humans. And then you have a duodenum, jejunum, ileum, colon. And what you are looking at, this dark color is actually the sticky mucus, strongly attached. Okay? And it's very difficult to remove that part of the mucus. Whereas the other one, which is called loosely attached. And if you look at the loosely attached, and then the thickness increases as it goes towards the colonic tissue. Okay? Any clue why we have more or thicker mucus in the colon? If you look at the depth, so you can clearly see the loosely attached mucus layer is much thicker in the colonic area. Any kind of guess? Say that again. Oh, HCl secretion. HCl secretion, you are talking about here, right? But you are saying less in this area. No, colon we don't have HCl secretion. We get mostly in the stomach. He was saying that because you secrete a lot of hydrochloric acid, and in order to protect our epithelial cells, and we need that. One of the things, the microbial community in the gut, it varies from top to bottom, right? So I think I have one slide. That will give you some indication, because in the colon we deal with a lot of toxic byproducts, okay, because of the microbial activity. And also we have a large number of microbes in the lower part of the GI tract. Remember, early part of the GI tract or gastrointestinal tract, we digest food, right? A lot of nutrient absorption happens. And as the food migrates, there are metabolic byproducts which are often toxic, right? That come out as a feces. So in order to protect our body from those toxic byproducts, we need to have a good protective system, right? That will be my first instinct. Like if there are a lot of toxic materials being produced in this area, so that needs to be prevented from absorbing by the epithelial cells. So there will be more problem, right? So that's one way to protect our body against the toxic byproducts produced by a lot of those billions of microbes we have in the lower part of the GI tract. That makes sense? That's like a more general instinctive approach, but scientifically that makes quite a good sense. If you look at the human system, this is again because lack of information from humans. So this is a study, I think 2010, they published it. They're saying that if you look at the mucus layer, the thinner layer again, or the sticky layer, strongly attached, and you can see very thin, and then the colon have a large loosely attached mucus. So this part of the mucus, during infection, your body can lose it, right? You can tell me a couple of diseases where you see mucoid diarrhea, mucoid bloody diarrhea, right? Couple of diseases happens. One is Shigella, you heard about dysentery, right? And also the amoebic dysentery. So you see bloody mucoid diarrhea, means a lot of mucus being released from that. So essentially this mucus layer get wiped off. Okay? But they try to provide protection. It's a strong barrier against infective agents. Okay. So as I said before briefly, the goblet cell function and the mucus secretion affect severely. So if that secretion changes during innate immunity, because that's the one going to provide large amount of protection, right? And so changes in normal microflora. So if your microflora community changes, then the mucus secretion would be also affected. Harmful enteric pathogens, I already mentioned that. Some of them cause mucus to be decreased, or sometimes it increases. Nutritional or nutrient deficiencies. So like I was mentioning before, if you stop eating or starving, and your mucus layer goes down, okay? And the villi structure also shortens, okay? A lot of things happen. So if you do histopathology, sectioning, you'll figure out that our villi structure doesn't work properly under certain diseases, or especially the nutritional deficiency. It's dramatic. So in order to monitor the effect of either probiotic or nutrient or any kind of positive attributes or sometimes negative attributes, you could measure the length of the villus. Okay, how tall is it? It can shrink depending on the disease condition. Okay, so these are the factors actually play a role. And in terms of the mucus secretion, it's also, as I mentioned, it controlled or regulated by number of factors, whether it's a microbial toxins or your physiological conditioning, a lot of those factors. So here is an example. Lactobacillus may increase the secretion, whereas the Helicobacter pylori down regulates. Helicobacter pylori, because it likes to live in the stomach, it decreases. And so these are some of those factors you can study, and that affect the mucus secretion. And as I said, mucus provides a tremendous barrier protection. Means it is an important player in innate immune system. Okay. All right. So here, for example, I'll show you the MUC2 I mentioned a few minutes ago. In this case, we are looking at certain function, but you can stain them, and these are individual goblet cells. This is the encoded by MUC2 gene. Okay. So if you want to study that if the mucus secretion is affected by, for example, in this case, pathogen Listeria monocytogenes, you don't see much goblet cell. Okay, only few. These are like a cross section of a villi structure, like the finger-like structure I told you. And so if you take a cross section, this is what it looks like, round. And so this is mucus secreting goblet cells. But whereas if you have a probiotic, you can see increased number of goblet cells. So that's a good thing, right? We need higher amount of mucus being secreted by the gut, initiated by probiotic organism. Okay, that's a symbol of health, or say I would say gut health. Okay, this is experimentally you can analyze it. These are some of this. This is also number of MUC2 positive cells you can count, and then you can also graphically present the data. And this is what I was saying. This is the villi structure. And remember, I was talking about the crypt, or where the stem cell are originated. So these epithelial cells, as they move, including epithelial or goblet cells, they all originated from the crypt. They are part of the small intestine or large intestine. Okay, and this length from crypt to this structure, if you are under nutrition deficiency, this will be shrunk. This will be shortened significantly. Okay, and also the crypt depth. If you are healthy, your crypt depth would be much deeper. That's what we call it. But in case you are infected or chronic stress or whatever negative condition may be, the crypt depth would be shorter. And again, technically, you can stain them. In this case, if there is a severe infection, you see this intense color, means the crypt is very active. Right? Why crypt has to be very active? So these are epithelial cells in this structure. This blue, this is the nucleus, right? Everybody remember this structure we talked last time, right? And so what is the thing I was pointing out? If the cells die, they will fall off from the tip, right? So if you see one treatment has a active crypt, this is intense. It took intense staining. Okay, try to follow me. Question I'm asking you. Whereas in this case, the crypt actually not taking intense stain. The staining represent the number of crypt cells. Okay, so if you have a high intensity, you have a more active crypt. Okay, low intensity means crypt or the stem cells are not that active. What is the difference mean to you? Why area in the crypt is highly active? It is making more cells. Why it has to make more cells? Why the body needs to make more cells? Because there are a lot of damage happening somewhere, right? Are you seeing this? Try to understand the logic behind it. So if cells die, that needs to be replaced, right? So these are part of innate system. So they are infected by pathogen, right? So now pathogen come in, they kill these cells. What body is going to do? Your response is going to repair. That's its job, right? So that I don't get permanently disabled. The purpose is to replace those dead cells, because those cells are killed by pathogen. Are you following my logic? So that means if the cells die because of infection, as part of innate system, my task would be, I need to protect my body, I need to protect myself. So what body does is that since those cells are dead, I need to replace those cells, right? Because those cells have certain function in the body. They are absorbing nutrients, right? If your epithelial cells are non-functional, you are not going to absorb nutrients. So you are going to show signs of diarrhea, right? That's what happened. So as a part of the innate system, if the cells die on the top, they needs to be replaced. So in this case, actually this is a infected cell by Listeria monocytogenes, is killing a lot of those cells on the top, and so therefore the stem cell try to make more cells. Are you clear? Not your head, if you understand. If you don't, then I'll try to reinforce the idea. It's very simple, right? It's like a more soldiers die in the war, you send more people, right? So essentially that's the same phenomenon. And if the cells are not dying in one case, there is a probiotic happen to be there, it's not killing those cells, so therefore the crypt doesn't need to produce more cells. So the lot of diseases, in order to understand the enteric disease, the gut health we keep on saying, if you kill these cells, like norovirus, you heard about norovirus mediated diarrhea? Norovirus, you heard about it? Or norovirus, or called Norwalk virus, is one of those viruses causes vomiting and diarrhea. It doesn't kill people, but you wish you are dead. That's the feeling. It's like if you suffer from any enteric diseases, a lot of times you go to bathroom so frequently, you feel that's miserable, right? Or you are vomiting because continuously replacing or removing all the fluid from your body. And so norovirus is one of the examples. that kills off all of these cells on the top. Okay, a lot of viruses or bacterial pathogens, Salmonella, Campylobacter, E. coli, they can wipe out this lot of this epithelial cells. So your trapped cells would be continuously working to replace those dead cells. It's a part of innate system. So I'm trying to make you understand what is the importance of mucus, right? And then how those mucus secreting goblet cells or our epithelial cell barrier, how they keep us healthy, how they keep us protected. And so our system, these stem cells would be overactive if the cells die on the top. It's kind of replacing all the cells. Okay, so that's the easy way to take it. You can count them. And sometimes also some cells undergo death, like a programmed death, programmed cell death, or necrosis. Also you can measure that. And again, these are part of the innate system to provide protection against infectious diseases. Okay, so what else do we have in the gut? So I mentioned that epithelial cells or mucus secreting goblet cells, they provide protection, right? And so if you look at in general, these all together they are going to provide protection. And one of the other tissues, easy to remember, called gut associated lymphoid tissue, or called GALT, G-A-L-T. So there is a GALT, there is a salt, there is a malt, mucus associated lymphoid tissue. So whenever you heard this terminology, easy to remember, think about it, very simple terminology. Gut associated lymphoid tissue. Lymphoid tissue, when you say lymphoid tissue, means it involves lymphocytes, okay, which is part of the adaptive immune system I mentioned last time. So they will be here. So this is actually the communication, or excuse me, the collaboration among the immune cells. So throughout our gastrointestinal tract, we have this in places. Okay. So one is, we talked about antigen presentation last time. You heard about that? Antigen presenting cells. So imagine somebody got in here, or let's talk. Say like if you bring some fruits and vegetables, you bought it from a store, right? And some product you cannot eat it raw, right? Okay, so can I have your attention, please? Okay, so I know everybody has a tendency to drift a little bit this way or that way, if something happening in the room. Anyway, okay, so just let me explain you a little bit briefly. Very simple way. You went to a grocery store, right? You bought some meat, vegetables. I should not say not veg stuff. Maybe say vegetarian stuff, right? So bought some leafy green, okay, and you decided to eat, right? So what are you going to do? You're going to cook, right? So you need to cook it, and then say you have a guest in your house, right? And then you're going to offer that to the guest, right? So you cook it, you offer it, right? And then at the end of the day, you are asking your guest to compliment you how well you cooked, right? Everybody clear? This is a very simple analogy, right? So your guest is happy, and he complimented you, telling you that how well it was prepared, right? And so in turn, what do you think? I feel good, right? I feel strong. I feel mentally, physically feeling strong. So if anything happens, I can defend. So you brought raw material, you cooked, right? You gave it to your guest, whoever came in, he complimented you, you felt good, then now you feel energized. Do you? You feel good, right? If your guests come in and tell you, oh, it's an awful preparation, right? I don't like it. Then you will be very disappointed, right? Are you with me so far? Okay, I like to see you nodding your head. Okay, so what happens in the immune system, exactly same thing happens, right? So if you bring raw materials, like if I catch a bacteria, right? I cannot process it, I cannot eat it right away, okay? So the bacteria needs to be cooked or processed, right? And then in order to get a positive response from my immune system, and then immune system needs to recognize that you are a good host, right? And once you offer that to the immune system, and then in turn they will complement you in the form of the antibody or cytokine, okay? That in turn you make you feel good. So what happens here is the collaboration. So if bacteria or pathogens come into the system, right, and then macrophage or dendritic cells, they are going to take that in, and they literally cook it, break it down into small pieces, and that's what we call antigen presentation. Okay, or those cells which present it, we call antigen presenting cell. Who do they present? Who their guest is? So let's say helper cell, T cell, T lymphocyte. Okay, and then they are going to collaborate with the B cell, for example. So the T cell can produce some cytokines because they are presented antigen, and then they can in turn activate the macrophage. So you're being a host, you feel now energized, right? And then you can go back and cook more, or if there is a pathogen coming, you can kill it better. Okay, and so the purpose of this lymphoid tissue, essentially raw material or pathogens coming in, they present it to the T cell, and then T cell and B cell communicate. The T cell can produce cytokines that can activate macrophages, they can kill themselves, kill the pathogens. Or if it is a cytotoxic T cell, there is another form of T lymphocytes that can also inactivate. And at the same time, if the B cell is activated, depending on what type of product you brought it from the store, and B cell may think that I can do a better job in helping, so they could produce antibody. So this is the lymphoid tissue. You will see that throughout your immune system, whether in the gut, whether in the mucus, whether in the skin, all of those different places, this lymphoid tissue play a role. Everybody clear? So the lymphoid tissue means adaptive immune response. So even though I was talking about innate system, I jumped into the adaptive because this is taking place all the time. And you heard about fire spats? Anybody? Some of you had heard that, right? So the fire spats is one of the localized tissues. You'll find it in the small intestine, and not in the large intestine, mostly in the small intestine. And that's where the M cell is located. And that's where the pathogens can, or microbes can enter the body, and then the immune system can play a role in controlling these. Okay, so this is how the lymphoid tissue look like. And then there are a lot more stuff in the mucous membrane. So we're talking about, these are the different components: lactoferrin, lysozyme, lactoperoxidase, a lot of antimicrobial peptides, as you mentioned. And these are all part of the mucous membrane. Okay. Since you already have the slides, you can read it more. One of the things I mentioned, that innate system, we cannot really induce it, right? But we do have a recent trend that we can train them. This is called trained immunity. Okay, and so this is kind of fascinating, that if a macrophage recognize a pathogen, right, and then maybe a week later or 2 weeks later, that macrophage again saw the pathogen come in, can it do its job as a first responder? Think about it. Usually macrophage or neutrophil, they will come into the site, they will try to engulf it, they try to destroy it. But before, we never thought that same responder, first responder, may not be able to do the job because they could be inactivated by pathogen. But what we learn now is that some of those macrophages, they can be trained. Okay, and that's where the trained immunity comes. So these are, I think it's a small printout, so may not be able. So some of those naive cells, it could be activated, right? It could be primed, means they already seen that. They could be trained, and they could have a tolerance. So this is kind of like a recent years new concept, saying that immune system can be trained in a way they could do a better job. Okay, I used that analogy before. In case there is a fire, the first responder come in, right? So if the first responders fail, then you look for backup, right? And then backup would be like adaptive immune system come in and helping. But in this case, those first responder, if they are well trained, they can do much important job even next time when they see it. Okay, so that's a small difference, but this concept actually proved to be very important and in communicating with the immune system and try to prevent it. So there is a resting phase, and then the second stimulation, and that's when they become more active. Okay, all right. There are a lot more about this trained immunity. I don't want to spend too much time. All the slides are there, and including the reference. If you want to read up that one, this reference, and it will give you, some resources for your knowledge. Okay, all right. then I talked about the resident microbes. So resident microbes also major part of the innate system, right? Whether the skin microbes I mentioned earlier, also the gut microbes, microbial community, and you have large populations in your gastrointestinal tract. So we have a bacteria, then we have a fungi, then you have archaea, right? All the yeast. So we have all those different organisms, parasites, also part of our microbial community, viruses also. So they are part of the gut community, and all of them has certain function in the body, and they play an important role in part of the innate immune system. So if I break it down again, I don't want to spend a lot of time, but the firmicutes, proteobacteria, bacteroides, and large number, right? So whenever you heard about proteobacteria, if you have too many of them, which is undesirable. Most of the pathogens belong to proteobacteria, okay, at the phyla level. Whereas the firmicutes and bacteroides generally very good, you want that population to be enriching your microbial community. And this gives you some numbers. So if you start with the upper part of the GI tract to lower part, so stomach or duodenum, stomach has a high population. Even though we have acids, we still have a microbes in the stomach. Then the small intestine, you will find anywhere up to 10 to the power 8 colony forming unit, or number of organisms, culturable organisms or non-culturable. Ileum and colon, anywhere close to 10 to the power 14 microbial numbers. So we are talking about very large number of the microbes in the colon area. So remember, now you can link that to mucus we talked about. Because of this such a large number of organisms, plus the metabolic byproducts accumulates in the colonic area, and that's why we need more protection. That's why mucus provides tremendous amount of protection. Okay. All right. So role of the microbiota in immune system is enormous. Okay. For example, they will produce gastrointestinal peptides. They also control endocrine cells. Short chain fatty acids, I mentioned that before, part of the metabolic byproduct. They are also important in the gut health. General function, xenobiotic metabolism, some kind of drug or drug molecules, toxic molecules, microorganisms help repair them. Also angiogenesis, new blood vessel synthesis, microbes help them. Help develop immune system. And then together microflora and immune system are very important. Remember the villus length and crypt depth I mentioned a few minutes ago. These are actually controlled or regulated by healthy microbial community. So now you see this how all are linked together. In immune system, gut microflora. I know we are going to have more discussion or more presentation on this, but then you can see this. These are major part of the innate and adaptive immune system. Okay, there are also a lot of other factors. Again, I want to give you the bigger picture. These microbes play other roles. Okay, but the negative effect of the microbes, we understood that if you have inflammatory bowel disease, right, you have a wrong population of microbes, they may have a negative impact on health. Okay, so those are some of those negative effects. All right, so a diverse community in gut early in life promotes a balanced immune response and prevents allergic diseases, obesity, diabetes. I guess you must have heard about the baby born by C-section, cesarean section versus the natural birth. Already most of you know that difference. So saying that natural birth, you get a lot of microorganisms from the mother that help you establish, colonize a healthy population in the gut early in life. The babies born by cesarean section has mostly a lot of different types of pathogens or microorganisms coming from the environment or the people who are handling, right, hospital environment. So there is a obviously huge difference between the babies who grew up with the natural microbes versus the organisms which are not so healthy population. Okay, and therefore that may dictate type of disease you suffer throughout your life. Okay, and that concept needs to be seriously used or practiced, because this is very important. It's like a wake up in the morning, what do you eat? Breakfast, right? So always you think that the breakfast, healthy breakfast, good breakfast can lead you a beautiful day all day, right? You believe that? So you don't want to get out of home or come to class without a good breakfast, right? Healthy breakfast. Think about the life. If you take the life, if you live 90 years and the 90 years equals to your one day, and how do you want to start your day? Think about it. With healthy, hearty breakfast, that's what you call it, right? And then you will see that your day going very well all the way to the evening and night, right? And so life, you want to start that way. And that can dictate a lot of different diseases. If you follow these recent literatures, the healthy, balanced microbial community makes a huge difference in someone's life. And as I said, a lot of diseases are linked to gut, so how your immune system responds, that makes a big difference. People suffer from asthma, right? Obesity, a lot of those different chronic diseases we suffer from. And a lot of this may start with the microbial community we start with. Okay? Keep that in mind. Again, you are not learning just for the sake of learning. See if you can practice it also. Right? That's the advantage of the students thinking, if I can save myself, right? If I can help myself. Okay, so that's my advice. If you look at the immune system in the mouth, throat, and respiratory tract, if I collect them, all of them together, so we talked about what we have in the mouth. Think about it, right? Saliva. We have a lot of antimicrobials, so lactoferrin, immunoglobulin, and then lysozyme, resident microflora, ciliated cells, that's in the respiratory tract, epithelial cells. So epithelial cells again come back to the how long they live, right? 10 to 12 days, don't forget. So that time also if pathogens attach to epithelial cells, some of the cells has a tendency to slough off, right? The bacteria go with that. And so that's also a immune strategy, right? Coughing, you expel stuff, and alveolar macrophage in the respiratory tract that help prevent pathogens, right? So now if you look at the biofilm in the mouth, right? Think about how your immune system is going to work. Okay, so this is essentially in the mouth. We start everything in the mouth, and if you look at of course that's doesn't look healthy, and you can see this inner part of it. You can see a thick biofilm, right? What about this? It's a biofilm, right? Because it's stained with blue, like a crystal violet staining. You learned that in the class, right? Crystal violet staining can stain the carbohydrate in the biofilm, and you can clearly see this is not a healthy teeth. So then you look at the lungs, right? And we talked about the cystic fibrosis lung because Pseudomonas aeruginosa grow in the lungs. So these are some of those biofilms in the mouth and the respiratory tract, right? So those are the problems there. Then how our immune system going to protect us against those microorganisms? We'll talk about that towards this lecture. Then in the stomach, right? It's pretty common now. You can tell me what we have, right? Acid, resident microbes, then enzymes, and also these are the healthy probiotic organisms, right? One of the conditions called achlorhydria, I think I mentioned that a few times ago. So although we know we have a healthy hydrochloric acid secretion, but some patients, they may not secrete hydrochloric acid. Maybe whatever condition may be. If you are staying up all night long because of the final exams, right, your HCl secretion would be diminished significantly. And that makes you vulnerable to other diseases. So that's also one of the terminology you can see, achlorhydria, and that makes you more vulnerable to infection. Okay, so the HCl is important in its system. In the small intestine, by now you should know the bile salts that play an important role. Especially it kills the gram-positive organisms, right? Then mucus, Paneth cell I mentioned a few times, gut-associated lymphoid tissue in the small intestine, and nasal flora. So these are part of the immune system protecting you against the invading pathogens. Large intestine, you're talking of the microflora, good microbial community, and also the mucus, as well as the short chain fatty acids. They play immunity. So all of those things are in place to provide protection against pathogens or invading agents. So if you look at the biofilm in the gut, again same mechanism I mentioned, right? It's pretty. So we have in the lumen, we are looking at the mucus layer in the epithelial layer, and you can clearly see this the attachment, microcolony formation, then maturation and dispersion, right? And so the natural microbes in the gut, they are forming microbial community in the form of biofilm, right? And these are healthy, right? This is what we expect. If you have a diverse community, they can provide good protection. But it's not always going to be healthy, right? If we started abusing ourselves with the wrong diet, wrong type of food, or different type of lifestyle, and so pathogens can, or some organisms can colonize, form biofilm, and then can cause chronic diseases. We all agree? So if you look at this, this is the microbial community we talked about, right? And if you have this disorders, so you can see this associated gut disorders associated with biofilm. So we are talking about irritable bowel syndrome, inflammatory bowel disease, peptic ulcer, and achlorhydria I mentioned, gastrointestinal cancers, and these are the type of microorganisms play a role. So we are looking at, we have displaced our healthy microbial community with undesirable pathogenic microbes. Now they are forming biofilm, okay? And once they form biofilm, and they are part of this chronic disorder, right? And so that's what you are going to see. The mucus layer disappears. These organisms become more invasive, right? They continue to irritate your immune system. So there is a influx of inflammatory cells. They're producing a lot of cytokines. So then more immune cells are coming into the site. And they are trying to protect against this invading biofilm. And now you made a complicated disease, which is the inflammatory bowel disease or chronic diseases. That also can contribute to the colorectal cancer, GI cancers, and that also become part of the immune disorder. Okay, and so biofilm play an important role causing those type of diseases. Here are some more examples. Again, these are new articles, and you are saying that if you are looking at the biofilm and you can see the redness, right? These are caused by some of those undesirable microbial community forming biofilm, right? And towards the end, of course, we'll talk about how to get rid of them. And I was using that example during colonoscopy. They can physically wash this or treat with certain antimicrobials or disperse the biofilm and then introduce antimicrobials, okay? Or also quorum sensing inhibitor molecules that can also break down. So number of different approaches people are using in the body to break down this undesirable biofilm, and that way you can help improve the health of the gut. Yeah, okay. And so the biofilm play an important role in these many chronic diseases. So there are more also examples. I just said therapeutic intervention against GI biofilm. So you want to inhibit the adhesion of these biofilm forming cells. You can also remove the EPS or eradication of mature biofilm. These are the different strategies they use to prevent or take care of this bad microbial biofilm in the body. Okay. All right. So these are other one. I think just said that here. You can also take healthy microbial community, right? The fecal transplantation, which is one of the therapy for C. diff or Clostridium difficile infection, and it's already approved, and I think some cases it's been used. Also, you can use healthy probiotics, right? You can use probiotics to replace the bad organism to help restore your microbial community and get rid of this negative effect of the biofilm. Okay. So this is the organ-wise how the innate system help protect you against whether any infectious disease, chronic disease, or chronic disorders. Okay, and so that's always working in place. So now think about how do the immune system recognize these invading pathogens, right? And one of the way we try to recognize is that How do I recognize you? Face, right? We have face, this is a feature. Everybody has a certain feature, right? And this is by your face, I know this is you, right? And this is how I recognize you. So now, immune cells, they are all sitting there, right? Macrophage, neutrophils, or dendritic cells, all of those cells, or epithelial cells. So the question is, how do they know which pathogen, which are non-pathogen in the body? We got a lot of non-pathogenic microbes, right? What is the best way they can recognize it, right? So they will say, well, he has the face, he got a hair, he got an arm and leg, and all kinds of structures, right? So the immune system try to recognize you by this feature on the individual, on the microbes, right? And so that's why initially we started with called toll-like receptor. We have a receptor for recognizing certain molecules on the cell, right? And these are different types of recognition system and then activates. So if you talk about that called toll-like receptor, initially it was discovered in fruit fly, okay? And what they recognize is that the epithelial cells, they recognize some molecules from the pathogens or microorganisms based on some of the key molecules, key features. So for example, flagella, right? So the epithelial cells can recognize flagella produced by any organism using one specific toll-like receptor or molecule, right? So that's specific. So if I say TLR5, toll-like receptor 5, it always recognizes flagella. One thing it cannot differentiate if flagella come from different organisms. They take it as like all flagella. So any organism with the flagella they help them for movement, right? And then TLR5 would recognize it. That's just the recognition part. Then we'll talk about the signaling cascade. So what it does actually, there are 1 through, I think there are 11, 12, NOD1, NOD2, and so what you are looking at, these different types of cells, whether it's a dendritic cell or monocyte or macrophage, a natural killer cell, or epithelial cells, they have these different types of molecules, okay? And it's telling you that if it is a flagella, TLR5 binds to it, right? If it is LPS, lipopolysaccharide, it's by TLR4. And so TLR2 usually peptidoglycan or lipoteichoic acid. These are the molecules in the cell wall structure I mentioned. And if you look at the other molecules, let me show you the graphics first. So here, think about this is one epithelial cell or macrophage. Okay? So it has this receptor molecule sitting on this. And this is like a PAMP molecule, pathogen associated molecular recognition, right? And so if these molecules are sitting, then it can bind to these specific antigens present on the microbe, right? So this is just the initial recognition. Why we need that initial recognition? Because you need to, if you want to get into a fight with somebody to kill it, right? You need to grab hold of that person also. It's not we are shooting with a gun or some other weapons, right? They will talk about that immune system, how they use it. But in this case, a lot of times it's like a physical hand fight, right? Both of you got angry and you want to, grab his hand and then twist his arm and all kinds of nasty stuff, right, to defeat that person. So essentially, these macrophage or dendritic cells or epithelial cells, they're going to grab hold to that structure. Once they bind to it, and next phase would be that you try to say, maybe you want to swallow it, right, or internalize it. So if it is a phagocytic cell, it will grab, take it inside, and then destroy it. That's the way it works. But the initial recognition starts this way. Everybody clear on this? Okay. Did you notice that some of those TLR molecules are inside the cell? So this is nucleus, nuclear membrane, and you can see this TLR3, 7, 8, 9, and also NOD2 and 1. These are all inside. How does it work? How do they recognize the organism? They are coming from outside, right? So when do the cell use these receptors? Are you following my logic? Usually if I'm in the intact cell, right, and my receptors are sticking outside, any pathogen or microbes come in, so I'm going to make an interaction, right? So now I can grab that and internalize, or I destroy it, right? But some receptors are located inside. How the immune cells using those receptors localized inside the cell? Anyone? How and when they use that? Those are for, not all of them, these are, those who are outside, these are called extracellular bacteria or microbes, right? But they need, some microbes like to get in, right? Intracellular pathogen, right? Or once the epithelial cell, the macrophage or dendritic cell, they bring the antigen inside, now this is another way of processing them. Now these receptors can bind to, if you look at those where they interact with, nuclear material, right? The RNA or DNA, they interact with this. So that means the cell, when they get inside, when they lyse, their nuclear material can bind to this receptor. Everybody clear on this now? You are bringing them inside, and then now you want to react against those organisms inside the cell. And naturally, a lot of intracellular pathogens there, right? Viruses are intracellular, right? They get in. There are intracellular bacteria also, they get inside the cell. And those organisms can be recognized by this TLR. Okay, now this is recognition. What is next? Now you recognize it, right? You take it in, you break it. Again, it's part of your immune system. You are trying to inactivate those organisms, you don't want it. Okay, so one of the steps of this interaction can lead to activation of signaling pathway. Okay, what is the signaling cascade? What we are looking at is that, what am I going to do? If I'm a macrophage, I grabbed on one pathogen, right? And what step come next? And next step is that, is it going to activate, say this is NF-kappa B pathway. NF-kappa B is, you know, the regulatory pathway, and that leads to activation of cytokine or inflammatory cytokine. Everybody clear? So this is the activation, and when you have these inflammatory cytokines produced by the macrophage, remember the kitchen experiment I was explaining to you, you brought food, you cooked, right? Offered to the, so what we are doing also during the time when you're preparing it, these cells can produce this inflammatory cytokine. And the purpose of the cytokine is that it can activate itself, or it can activate the neighboring cells which are nearby. And they, if they get activated, they can all come in, now destroy the pathogen more effectively. Is it clear? So inflammation, you heard about inflammation, right? And most of the time we talk inflammation is really not desirable, right? But immunological sense, inflammation is an important step. So there are degree of inflammation, you can say, right? So if you have a massive amount of cytokine produced, you can have a massive inflammatory response. That's the bad part. You don't want too much immune cells to come in, and they're going to cause more problem, right? But in this case, inflammation at low level means lower amount of cytokines produced, and that can activate immune system. And then you need that immune cells to come in and help remove those invading microorganisms or toxins, whatever causing that inflammation. Everybody clear? So at low level. So NF-kappa B play an important role. This signaling cascade, the nuclear factor get into the RNA, excuse the DNA, it binds to the promoter, and it activates the cytokine synthesis. Cytokines are proteins, right? Whether it's a TNF alpha, IL-1, 6, name any of the cytokines. Got it? But mostly inflammatory cytokines we are talking about. These are two different, one is called inducible, one is constitutive, means always happens, other one is the inducible. Microbes, depending on the microbes or the agents, it can do this. Remember, whatever immunology system you study, this is central. This is always going to happen. Okay? Alright, so I think that's a good place to stop. Any questions on this? I know, I mean, this is supposed to be the last part of the immunology, but as I said, I think afternoon lecture I will also continue this so that you get a global picture. So the innate immunity, right? We talked about innate system, what is in place, right? How well they can provide protection against inflammation, or excuse me, invading pathogens or microorganisms or toxins, which is undesirable, right? So do you think this is, we're talking about the negative aspect, right? Negative part of it. Do you think any good thing also induce inflammation? Good, say for example, good microbial community or probiotics. Well, autoimmune disease, I think you're talking about, that's a little bit different. Okay, there are different aspects of it. But in terms of the inflammation, inflammatory conditions, usually activated by undesirable pathogens, or which is negative. Usually, if you say probiotic, usually it has a called anti-inflammatory response. It doesn't promote inflammation, but there would be certain components. If you take, for example, peptidoglycan, I mentioned that, right? Probiotic has that peptidoglycan. They could have also teichoic acids that also can induce or activate. So it can, some situations that can do it, but the degree of inflammation or inflammatory response is much lower than the pathogens. Okay. So is inflammation desirable or undesirable? We just mentioned, right? So desirable at low level. So it's not bad if you have certain level of inflammation, because you need to recruit other immune cells to get activated, right? And they can perform better. But if you have a severe inflammation, that means we are producing massive amount of undesirable immune cytokines, right? And that can cause severe disease, right? Including septic shock. I mentioned that a lot of times that the severity or fatality happens because our immune system is overactive, right? COVID is one of the examples. Some people die because of the aggressive response by your immune system, producing large quantities of inflammatory cytokines. So there is a degree, there is a requirement. So if you are doing an experiment in the laboratory, so now you have to define actually if certain treatment showing certain level of inflammatory cytokines, is it good or bad? Because you have to measure it in relation to the overall health of that individual or animal. Okay, and that's how you have to interpret the data. Okay, any questions now? You have the microphone? Or no? Any questions before you go for break? You should have a lot of questions. You studied the slides, right? Okay, go ahead. Oh.
Sir, let's suppose there is a biofilm specific antigen. So can we generate memory T cells against it?
Biofilm specific antigen and memory response against it?
Yes, sir.
Sure, you can do that.
Sir, if we can do, then why do immunity fail upon the reinfection?
Is there a chance of reinfection if you do that?
So then why do immunity fails when we got reinfected?
Sure, if you have a memory cells and your memory cells can work against that if you have a reinfection by same pathogen or same microorganisms producing those biofilm-like molecules. It can happen, but then again, all depends on the degree of infection, the severity of infection, how well it's going to work. You got to think about the biofilm. We are looking at the community, right? A structural aspect, and there is we'll examine towards the end. There are different layers, right? How immune cells going to reach to each layer, and that's the most limiting factor. If the immune cells can easily reach to the surface layer, for example, right, which is easily accessible. If the cells are inside, or the exopolymeric substance is there inside, even though you have a memory cells developed against that antigen, but immune cells may not be able to reach because of the physical barrier is already created within the structure. That makes sense? So
Thank
it
you,
will
sir.
work from the surface. So that's why it is effective against individual planktonic cells, but when it comes to biofilm, it's a whole lot of different game. And that's when it will become, somebody said that before, it will become chronic infection, right? It's not acute. So the best way to get rid of it would be, you need to disperse the biofilm, make it individual cells, and then immune system can work very well. Okay? So it's just the biofilm when it is formed and how effective your immune system would be, that's problematic. And that's when it becomes chronic disease, means a lot of times these biofilms are formed and they are continuously present, and your immune system try to do a lot of stuff. And they wrap it up, they cover it, they form granuloma, they form cyst-like structure, a lot of those things. These are actually containment of the infection. That answer your question?
Thank you, sir.
Okay. Any other question? Oh, there's a question. Go ahead. Oh, you have to run other side.
So please can you re-explain that part where you explained the toll-like receptors, how they differentiate between probiotics, like good bacteria and bad bacteria, pathogenic bacteria?
That's a really excellent question. So what you know so far, even though peptidoglycan structure, for example, right, and is present in gram-positive organism, all gram-positive organisms, but there will be certain molecules or structure that may present something different. But if you study the probiotic literature, sometimes people complain that some probiotics can induce inflammation. Okay, so probably those probiotic organisms are recognized by immune cells through TLR receptor, like whether the peptidoglycan or lipoteichoic acid or wall-teichoic acids, they will be recognized and they are going to activate immune system. So in that case, you will not be able to differentiate it. But the degree of severity of the response may vary from probiotic to pathogens, definitely. The pathogenic response, you got multifaceted. That recognition followed by immune cells, they're going to produce more stronger response. Because remember, pathogens not only have the structure, they got all those other virulence factors. They got a surface adhesion or pathogenic factors, some of them may be producing certain toxins. All of these different molecules, they are produced by the pathogens. So therefore, your immune system response is going to be much more severe against that compared to probiotic. It's only just maybe peptidoglycan or some of those components, but it doesn't have those pathogenic factors. Okay? And therefore, your immune system is not going to aggressively respond to that. So this is how immune system play a role in differentiating this, which is a bad guy versus which is a good guy.
Okay, thank you, sir.
Okay. Any other questions? Okay, looks like you are ready for the break. I think you can have coffee or tea, then that will help you wake up. So come back and we will have a guest speaker here.